Hi, this is Tara Saxic, Bachelor's Prepared Registered Nurse at Liberty University. This is MSCI 520 with Dr. Ben. I'm going to be presenting on disease pathophysiology of muscular dystrophy, understanding the cellular pathophysiology of muscle degeneration. We'll start out with an introduction of what muscular dystrophy is, and it's noted and defined as a variety of inherited neuromuscular disorders characterized in a group by progressive muscle weakness and degeneration. So on the cellular pathophysiology level, these muscular dystrophies predominantly involve proteins that are crucial for maintaining muscle cell structure, integrity, and function. And what happens is these specific proteins end up mutating, and with these alterations, damage of muscle fibers occur, as well as inflammation being triggered, and then eventually it leads to muscle wasting, which is also known as muscle atrophy. There are different types of muscular dystrophies, which are also caused by mutations in various genes encoding proteins that are involved in muscle cell function, and also the basement membranes of the extracellular matrix. Basement membrane is synonymous with the term basal lamina, which you may hear later on in this presentation. We'll refer to types, epidemiology, and also etiology of muscular dystrophies here. This is different forms of muscular dystrophy would include the most common form, which is Duchenne. Duchenne is where the dystrophin protein is completely absent. Now there's also a form of muscular dystrophy called Becker, and this also has a consequence of the dystrophin protein as well, but it's a milder form because it's either just a partial loss of it or a dysfunction of it. But for this presentation, we're going to focus on the morosin deficient congenital muscular dystrophy, also known as MDC1A. This primarily involves the disruption of the muscle cell membrane stability due to the absence of lamina alpha 2 protein. This is encoded by the LAMA2 gene. This form of muscular dystrophy is classified as a subtype of congenital muscular dystrophy. So in 2021, the NIH had noted that this specific type of muscular dystrophy affects 1 in every 100,000 to 1 in every 500,000 individuals. Even though it's considered rare, this form accounts for 24 to 37% of all congenital muscular dystrophies and has the epidemiology resulting in a higher instance where consanguinity is more common. So consanguinity is inbreeding. So bloodlines do matter whenever it comes to muscular dystrophy and the diagnosis of it. Normal physiology and homeostasis. So like with everything, this is the grand challenge to physiology, is maintaining normal physiology and then also acquiring ongoing homeostasis. So easier said than done in most cases. So in normal physiology and homeostasis, thermoregulation, blood pH, fluid and electrolytes, glucose levels, the balancing act of oxygen and carbon dioxide, and also the endocrine biofeedback loop. They all play a heavy role in normal physiology and homeostasis. The body is constantly in flux, adjusting to its internal environment, which includes balancing numerous bodily systems and processes to ensure essential functions are performed. So here we're going to look at the pathophysiology and molecular basis of the specific MDC1A. The pathophysiology here of this looks at the deficiency, or more likely the absence, of laminin alpha 2. This is a critical protein that enables structure, integrity, and function of the muscle fibers. In muscular dystrophy, particularly in cases related to this laminin deficiency, laminin is labeled as a grouping of glycoproteins, which the basal lamina, otherwise known as the basement membrane, as we mentioned earlier, of the muscle tissue is the crux of its assembly. When this becomes disrupted or reduced, it leads to impaired muscle cells and adhesions, increased susceptibility to mechanical damage, and then ultimately muscle degeneration and weakness. Now, this particular disruption often occurs due to the mutation of the laminin alpha 2 chain. And this is a key component of the laminin complex in muscle tissue. The laminin is made up of the alpha 2, which you can see there in the slide, specifically in the picture. Laminin looks like a cross, which we will discuss more later, but you can see that it is labeled alpha 2 chain. The beta 1 would be the blue arm, and the gamma 1 would be the orange arm. And these are subunits that are intertwined, as you can see, going down the spindle of the cross in the center to the base. And these are held together with disulfide bonds acting like molecular glue. In the schematic, as you can see there, it shows the alpha 2 as well as the laminin 211. A laminin 211 is another name for morosin, which is specific to this type of muscular dystrophy that we are talking about here. And so what happens here is the LAMA2 mutates, resulting in the morosin deficiency in the extracellular matrix of skeletal muscles. The cellular mechanism with laminin, it interacts with specific receptors on muscle cells like integrin, alpha 7, beta 1, which are also very critical to cell adhesion, migration, and then also signaling pathways necessary for muscle function and regeneration. The consequence of this disruption, though, is that the basal lamina, also known as the basement membrane, is comprised because it's compromised, should I say, because of the laminin deficiency. So what happens is these muscle cells lose their adherence abilities, dissolving their ability to attach properly, thus making cells very vulnerable to damage from a host of different factors, such as mechanical stressors. And this would lead to muscle fiber breakdown and then also fibrosis. Now, differentiating between signs being observable and symptoms being subjective, typically this is diagnosed at birth or early childhood. So at birth and early childhood, like birth for sure, symptoms would be kind of vague. You'd have to go more towards the signs. And signs would indicate hypotonia, motor delay, and failure to thrive. As the child might get older, there might be more symptoms that they would be able to voice and complain about. And things would also be noted. At sometimes signs and symptoms can be interchangeable, such as contractures being a symptom, muscle weakness, and then also sensory impairment. Now, for laboratory features and results, you'll see that the two enzymes there, the creatine kinase and then also the aldolase, they will both be elevated when testing for MDC1A. Also noted would be MRI abnormalities down the road, such as muscle atrophy, fatty infiltrations, fibrosis, and then also muscle edema, which is swelling. An EMG could respond back with decreased motor unit potentials as well as increased spontaneous activity. And muscle biopsies would indicate degenerating, regenerating muscle fibers, fatty infiltrations, also noting the fibrosis, absence also of the laminin alpha 2 on immunostaining. And then lastly, of course, molecular genetic testing. This would denote the mutations in the LAMA2 gene, and which would be detected by sequencing or deletion analysis. Now here you see a host of different differential diagnoses. Other congenital muscular dystrophy types, obviously myotonic dystrophy type 1, spinal muscular atrophy, mitochondrial myopathies, glycogen storage disease, myasthenia gravis, and Charcot-Marie tooth syndrome. Complications and prognosis when it comes to anything revolving the muscles, we are comprised, a lot of our organ systems are comprised of the functionality of muscles. So musculoskeletal, cardiac, GI, respiratory, psychological, because, well, this is going to play heavy on the psychological aspects of a person. Also immune, is the immunity is compromised, but also pain. Pain, with the inability to move. Then, of course, the fibrosis and the contractures coming into play. And again, all of this stuff stemming back to the psychological component. Prognosis varies depending on the severity of the disease itself, the age of onset, specific genetic mutations that are involved. Motor function progressively worsens, and the probability of complications can decrease the lifespan of these individuals. Now, one thing I wanted to notate is with the words building block, basement membrane, and knowing that laminin resembles a cross, it took me down a very big rabbit hole. So, um, looking at the Christian worldview concept here, the song Jesus is the Rock of My Salvation came to mind. So when I looked, knowing that laminin resembles a cross, I looked at what the rock in the song meant, and it's the meaning of faith. Our faith is built on Jesus. So the following line is also his banner over me is love. So I looked up to see if any molecular structure resembled the heart. Serum albumin has this characteristic. And it is noted that the BCAAs, the amino acids, are used in muscular dystrophy treatments. Albumin is comprised of 585 amino acids, which are can be broken down, and then, of course, reassembled into other proteins required by the body where deficiencies may exist. So next, I looked up other words correlating with Jesus and the foundation or rock, and I saw the term cornerstone, and that stuck out to me. So again, I researched if there was a protein that resembled the cornerstone in the body, or was it, or something that correlated with human functionality, and was led to the term pluripotent stem cells. And it kind of blew my mind because they are currently right now researching this particular concept for muscular dystrophy. This is because when these stem cells are reprogrammed from somatic cells within the patient's own blood, the process of these being reprogrammed is called induced pluripotent stem cells. They then behave as embryonic stem cells, allowing the development into various cell types. This would lead to less of a possibility of rejection as well. So these can replace damaged muscle tissue with healthy cells. This could potentially overcome the genetic defect, further proving that God is in the details and is most significant in finding truths in every place we look. Thank you.